So hello everybody, my name is Ambra Pozzi. I'm a professor in the Division of Nephrology at Vanderbilt University, and I'm the past president of the American Society for Matrix Biology. Yes, running and walking. It's my mental liberation of all the troubles in the world. The favorite molecule probably are the receptors for extracellular matrices, particularly integrins and discoid domain receptors. So I don't have a single molecule, I have two molecules, but they are linked to the extracellular matrix. A discovery that I made is that some of these receptors, as full receptors after binding the extracellular matrix, can translocate either to mitochondria or to nuclei as an intact full proteins and regulate fibrotic responses. This is actually something that we are very much interested in the lab. So I think it was an indirect way to come in because I was studying the integrins at the beginning, and I was studying collagen binding receptors integrins alpha 1 and alpha 2. Then I studied discoid domain receptors that bind both fibrillar and non-fibrillar collagen. So I'm basically affiliated to both the basal membrane group and the fibrillar group of the ECM collagen. So in the PhD, he was a wonderful person. His name was Ragnotti, and he was a pathologist, and he taught me really to understand the pathological aspect of diseases. And then I was lucky enough to have a young investigator. He was honestly my age at the time when I was a postdoc, and Humphrey Gardner, who introduced me to the world of integrins, the collagen binding receptors. And then when I came to Vanderbilt, I have different mentors, but one of them was probably Billy Hudson, that really for me is really my hero in the field of the ECM. Yes, I think that in addition, of course, to Billy Hudson, I would say that the fibronectin field was very, very big when I was really a postdoc. So like Herculus Nati, for example, or Richard Hines that was working on the ECM and looking at the receptors. These are probably like my two heroes. The major question is how really collagen or in general ECM is regulated in health and disease. It's not only one pathway, it's not only one disease, there are different pathways, different diseases. So I think that we should try to do a personalized ECM targeted drugs in a way, because as I said, it's more than one organ, more than one pathway. So to the younger colleagues, don't be afraid to try new avenues. So when we discovered, for example, that these receptors were going to mitochondria, at the beginning we were like, no, it cannot be possible. But it's because I had a very curious postdoc who actually we never gave up. Don't give up. There are no such things like silly questions, silly problems. One door closes, another one opens. Take it, and don't be afraid. So I think that the cool story for me was when we had for the first time 3 societies running a meeting together, and that one really happened a few years ago. And we had 3 societies, and it was actually interesting to see how their interest in ECM was tackled by different angles. But at the end, we really worked out a wonderful program in Salt Lake City, and I think that that one was really the demonstration that different groups can work together to really have a final wonderful program. How it's regulated, God. Tell me, please. Dogs. Collagen.